paper we have reviewed is on post-translational modification, and its title is Silencing of CXCR4 Blocks Breast Cancer Metastasis. The aim of this research was to determine whether lowering CXCR4 mRNA levels by using siRNA might inhibit breast cancer metastasis. Double-stranded RNA in cells is processed into the short 21 to 22 nucleotide strands called siRNA that are used in an in vivo therapy injection and can be detected within 24 hours post-injection in organs. Metastasis in breast cancer is a stage of breast cancer where the disease has spread to distant sites. Breast cancer cells metastasize to regional lymph nodes, bone marrow, lung, and liver in an organ-selective process. Chemokines are critical mediators of cell migration in the immune system. Chemokine receptors CXCR4 are integral membrane proteins, receptors for CXCR12, and critical factors for breast cancer metastasis. In an in vivo process, fragments of DNA are inserted into a vector to be amplified in a host cell, whereas in vitro, components of an organism are used and isolated from the natural surroundings. Inhibition of CXCR4 and CXCR12 interaction is done by selective antagonists, which blocks breast cancer metastasis in an in vivo process. Two different siRNA duplexes of CXCR4 were used in the experiment. siRNA 1 was designed, whereas siRNA 2 was purchased. 6 to 8 weeks old female mice were used in the experiment. They were divided into 6 groups with 6 mice per group. Group 1 was given injections of tumor cells transfected with non-specific control siRNA duplexes, and later given injections of control siRNA twice weekly. Group 2 and 3 were given IV injections through the tail vein of tumor cells transfected with CXCR4, siRNA 1 and 2 at 48 hours before the animal injection. Group 2 were treated with CXCR4 siRNA 1 and 2 twice weekly, whereas Group 3 were not treated after administration of tumor cells. Group 4 were given injections of tumor cells without prior siRNA 1 and 2 transfection and were treated only with CXCR4 siRNA 1 and 2 twice weekly post-injection. Group 5 and 6 received IV injections through the tail vein of tumor cells transfected with CXCR4 siRNA 1 or 2 at 40 hours before the animal injection and were treated with siRNAs twice weekly. Animals were sacrificed at 45 days after the tumor cells injection. Mice in Group 1 and 2 were put into a micropipette scanner prior to sacrifice. Whole lung tissues were harvested in optimum cutting temperature. The experiment was repeated to confirm the results. After 48 hours, the transfected cells were collected to measure the mRNA and protein levels of CXCR4. Reverse transcription PCR amplification was used on the lung tissues. Micropipette studies were done using 6 mice from groups 1 and 2.
Results. siRNA 1 was more efficient in reduction of CXCR4 expression than siRNA 2. The combination of siRNA 1 and 2 achieved even better expression of CXCR4 expression at the mRNA and protein level. The invasion of MDA MB231 cells transfected with siRNA decreased to 39% of the control cells, 51% with siRNA 2, only 16% with both siRNA 1 and 2. 45 days after the tumor cells injection, all the animals in the control group developed lung metastasis. Only one animal in Group 2 developed metastasis. The crude estimation of the average area of micrometastasis on the lung surface from 6 animals. Group 1 was 6.9% and Group 2 was 1.7%. 3 representative pictures of the lung from 5 treated groups show significantly fewer visible lung metastasis, most notably decreased in 2 and 5. The HNE, which is hematoxin and eosin staining of the lung tissues from Group 2, show the morphology of the normal lung. Control group shows invading tumor cells. There was significantly fewer metastasis in the lung of Groups 2, 3, and 5. The high CXCR4 expression in the control group mouse lungs and much lower CXCR4 expression in the lungs of the treated group.
siRNA 1 and 2 was specifically developed for the CXCR4 mRNA. CXCR4 suppression was more effective when using the two combined siRNAs than either siRNA alone, as combining the two enhanced gene silencing by decreasing protein and mRNA expression. Lowering CXCR4 mRNA levels by siRNA inhibits CXCR4 invasion, as was measured in the Madrigal invasion assay. Previous research has shown that using anti-CXCR4 or antagonists to block CXCR4 interaction reduce metastasis in the lungs. In this research, we used antisense technology to reduce CXCR4 levels instead of blocking receptors from interacting with ligands. This is more effective as prevention of metastasis by CXCR4 antagonists doesn't guarantee metastasis prevention. However, both methods were required to confirm CXCR4 inhibition prevents cancer metastasis.
Correlation coefficient. The correlation coefficient is used to determine the linear association between two variables. It will always vary from -1 to 1. -1 showing no linear association, and 1 showing 100% linear association. In this experiment, the value was 0.9674%. This shows that there is nearly 100% linear relationship between HHPRT and CXCR4. This shows that lowered CXCR4 expression levels blocked breast cancer cells forming lung metastasis.
In conclusion, RNAi treatment successfully reduced breast cancer metastasis. RNAi is more efficient as a method of treatment in comparison to using antagonists to block CXCR4 receptors. Reduced transmembrane CXCR4 receptors inhibits the ability of lung metastasis to occur. The use of controls, i.e. non-specific siRNA, confirms that specific siRNA complementary to base pairs of the CXCR4 gene was responsible for reducing breast cancer metastasis. Combinations of siRNA produce more efficient suppression of the CXCR4 gene. The lack of cytotoxicity confirms that therapeutic doses can be developed to reduce cancer metastasis. Human trials can be established as lack of morbidity or adverse effects during treatment.